Hello, everybody. I'm Ali, and Andros is also here. We're going to be just in the background, making sure everything's flowing smoothly. So let me just introduce our guests, our organizers. May I begin?
Yes, sure.
Luke and Stephen? Okay.
Sure.
So we have Dr. Stephen Declerck. He's a senior researcher at the Netherlands Institute of Ecology and also a part-time associate professor at KU Leuven in Belgium. And although having a background in metacommunity ecology and biodiversity research, he recently also has been doing research on experimental evolution in relation to ecological stoichiometry and ecological stressors, as well as on cryptic diversity in zooplankton. And Luc de Meester is the other organizer of this session, and he is the director of the Leibniz Institute of Freshwater Biology and Inland Fisheries in Germany. He's also a part-time professor in ecology and evolutionary biology at KU Leuven, Belgium, and professor of freshwater science at Freie Universität in Berlin, Germany. His interests are broad and include aquatic ecology, biodiversity and nature conservation, metacommunity ecology, evolutionary ecology, ecological genomics, eco-evolutionary dynamics. There were more on the list, but I kind of just edited to keep it shorter. And we are very happy to have you both here. We'll just be in the background in case anything comes up. So the floor is now with you, Stephen and Luke.
Okay, I'm going to share my screen now. Try at least. Do you see my screen?
Yeah.
Yes.
Yeah. Okay, then we can start. Okay. Thank you very much, Ali, for this introduction. Today's session is all about traits. Traits are key to ecology. More than just the taxonomic identity of species, traits provide us much deeper insight in how exactly organisms interact with each other and may also impact their biotic as well as abiotic environment. And this is true for individuals, populations, species, and even entire communities. It's also important to realize that the phenotypic traits of an organism are also strong determinants for its relative success, and also that plasticity of traits allow organisms to quickly adapt to a rapid change in their environment. And to the extent that trait variation is also genetically determined, selection on genetic variation may result in changes in the genotypic composition of populations and may allow populations to rapidly adapt genetically to environmental change. And a roughly similar kind of process happens also at the level of species, where selection on traits of species may result or may actually determine the functional trait composition of communities. And the fact that selection on genetic variation and selection on species traits may happen simultaneously opens also the possibility that both processes interact with each other, and this opens a whole array of potential eco-evolutionary dynamics, also potentially affecting ecosystem functioning. So the aim of the session of today was to attract interesting contributions that can illustrate the importance of trait variation in the ecology of shallow lakes. And we got a very nice and diverse set of contributions that illustrate the importance of traits, especially pertaining to two major levels of biological organization, being the population level and the community level. There will be three contributions that illustrate how traits can be shaped by the environment. There's two talks that illustrate the importance of trait variation for ecological interactions between species as well as for ecosystem functioning. There's three talks about how populations adapt to environmental change. 2 refer more specifically to rapid genetic adaptation, whereas another talk will talk about rapid adaptation in the absence of initial standing genetic variation. And then there's 4 talks that will illustrate the importance of functional trait composition and diversity for the aquatic communities. A few words about how we are going to organize this session. So each contributor will get some time to present his work briefly in a short pitch of about 2 minutes, helped with PowerPoint slides if they choose to. And this will be followed by a question and answer session of maximum 5 minutes. We cannot allow more time, otherwise we will it will take too long. But depending on how much time being used by the contributors, we may still have some time left for more general discussion. We will see. So I would like to recommend all the contributors for their very entertaining videos and also inspiring work. And both Luc and I cordially wish you a very interesting and inspiring discussion. So we will go now through the list of the different speakers, and I would like to invite Quentin Struik to start with his speech. Thank you.
Thank you very much. Let me see if I can share my screen because I prepared 2 slides for today. So my presentation was about the water hyacinth traits in controlling methane emissions from freshwater ecosystems. And we conducted this research because we found that there was a high variability in methane emissions among distinct geographic regions from water hyacinth beds. And you can see a water hyacinth bed here. So our objectives were to investigate the influence of plant traits, for example, biomass, surface area, and vitality, on the methane emissions mediated by Egonia crassipes, which is a scientific name for the water hyacinth. And the second objective was to investigate the role of key environmental factors that mediate these methane emissions. And our results in a nutshell were that we found that methane emission was increased significantly along with biomass, leaf surface, and bulb surface, but also with plants' volume and plant vitality, as well as with water temperature. So we assessed the plant traits as well as the environmental factors. And in addition, we found a causal relationship between methane emissions and the concentration of methane in the water that was around of the roots of the water hyacinth. And our results showed that interspecies trait variation can strongly govern ecosystem process rates. And you need to take this into account in order to accurately predict methane emissions from macrophytes and water hyacinth beds. So this was my pitch.
Okay, thank you very much. There was a possibility also to ask questions through the platform, through the internet platform, and I checked them recently. I hope since then no new ones were added. Maybe I can start with the question that was asked, and that was a question by Renske Vroom. I don't know if she is present here at the session now. No, if not, then I will ask the question from her to you, Quentin. So she says, a very interesting presentation, great work. She has actually 2 questions. First, she says, you mentioned the temperature difference in the young and old bulbs. How does that affect methane transport? Would it explain the difference in emissions due to bulb age? To me, it would seem that a bigger temperature difference would result in more diffusion. Is that right?
Okay, so indeed you would expect a bigger temperature difference, that it would lead to more diffusion, of course, due to the temperature gradient. And we found that the younger bulbs that were warmer, we found that the younger bulbs had a higher temperature, but also a lower internal methane concentration. And we think that this is because these young bulbs, which are more vital, emit more methane compared to old bulbs. And the old bulbs that were cooler had an internal, like a high internal water concentration, because these bulbs consisted of a lot of water. And since methane is a poorly soluble gas, there was like a barrier between the atmosphere and like the internal methane concentration. So the methane needed to pass through the water first, but since it's so poorly soluble, it chose to go all the way up to the young bulbs, and so you have methane buildup in the old bulbs.
Is there anybody who has a reaction or additional question about this aspect? No? Then I move on to the second question of Renske. And she says, besides the incubations, did you test methane emissions using the same setup, but with no methane dissolved in the water phase? Could it be possible that some breakdown processes may take place in this environment?
Yes, we did test this. We used different kind of waters, because we also used the nutrient water, but also tap water and rainwater to test this. But all these waters without the methane dissolvent did not reveal any methane emission. Yeah, in other words, there was no production in this system.
Okay. Anybody else of the audience here having a question?
Ali has a question, I think, and then
Yeah,
Pedro.
I don't know if I'm allowed as a host, but
Yes.
I had a quick question. Just curious if you guys measured how the emissions change over time.
No, we didn't. We placed the plant in a setup for 3 hours in order to make sure that there was a flux, and then we measured the methane emission 2 times, both for 5 minutes, in order to reduce insecurities. But we did not look at a change in a pattern over time. No.
Quentin, I
Okay.
don't know if you remember, but in the field it was during the day and during the night that the emissions were measured, and we found no significant differences.
No, and also in the laboratory when we used the light intensity, we also did not find a significant difference between shade, darkness, or light conditions.
Okay, I would say one question more, because we are running out of time already. Yes, Pedro.
Okay, I was just curious, Quentin, do we know anything about, I'll go back to some of, I think, Steve's question to my talk. Do we know anything about the genetic variation for those, and whether, if there is one, it would be interesting to manipulate, right, populations to regulate their methane emissions accordingly, because you can landscape those communities to do the tricks we would like them to do it, right?
Yeah, actually, I'm not sure. However, we use like an identical line, because we have these water hyacinths already for a long time in the greenhouse, and they reproduce asexually. So I assume that the genetic variation, at least in our experiment, was the same. They were all like clones, but I'm not sure how it is in the field. Maybe Sarian knows a bit more about this.
I have not seen any work looking at
Yeah,
that. So
that could be interesting. All right, thanks.
future work.
Yeah, thank you.
But clonal could be good for this case, to manicure landscapes with clones that are actually very good at.
You can change the clones. So before we go on, I sort of noticed, Quentin, from your video that you are still a master student. I want to congratulate you with your video, because for being probably the youngest amongst us in science, you did a very good job.
Thank you very much. Thank you.
Okay, then we go to the second contribution, and that's the contribution by Alice Dalla Vecchia.
Hi, everyone.
Yeah, she made a contribution about local patterns and drivers of functional traits in Nuphar lutea. I invite you to move
Yes,
your...
I'll try to share my screen.
Yeah.
So, can you see my screen?
Yes.
Okay, perfect. So, I am Alice from the University of Parma. I'm a PhD student, and my study was about the local patterns and drivers of functional traits in Nuphar lutea. So we wanted to employ a trait-based investigation to reveal the local adaptation of this species. And specifically, we wanted to quantitatively measure the intraspecific trait variability and explain this variability in relation to water and sediment characteristics. So we were working with specifically leaf traits, because we also wanted to demonstrate that the variability of leaf traits can be evaluated from the spectral responses of both the individuals and the canopies. And we did this in a shallow eutrophic lake in the center of Italy. So what we found was that leaf traits were actually visible from the leaf spectra, but the calculations methods are so far calibrated for terrestrial species, so they are not very accurate for some leaf traits for aquatic species. So that at least is what we found, and they may need some more calibration. And leaf traits did show a high variability among different plots. So even for one single species in the same lake, there was a high variability in the trait values. And it seemed that structural traits, for example, leaf area or weight, were more related to water depth characteristics, and biochemical traits like pigments were more related to sediment characteristics. And we also acknowledge the importance of special characteristics for the leaves of these species, Nuphar lutea, that needs to reach the surface in order to photosynthesize and to live. So this was what we found.
Thank you very much. Any questions? Luc?
Yes, thank you, Alice. So yeah, I have actually two questions. The first one is basically exactly the same as in the previous talk, of course. I mean, I guess you have no information on genotypic composition of the microphytes in the lake, and so it might just be interesting perhaps to check that. The second question is whether you tried with those different spectral compositions to also look beneath those leaves, whether there would be differences in productivity or something like that, whether that would have a direct consequence for the lake.
So for the first question, we actually will have information on the genetic variability, because there are three research units. So the first two are working on spectral and functional traits, and we still don't have the data, but the third unit is working on this genetic information. And so far, so the only thing we calculated for the lake is the chlorophyll. So it's a very eutrophic lake, and we don't find any submerged macrophytes, for example. So we don't have any plant productivity beneath Nuphar lutea. But we did calculate also the chlorophyll because there are very strong blooms of cyanobacteria and other algae.
Yeah. Okay, thanks.
You're welcome.
I have also a question. So you say at the end of the talk, when you explain the results of your mixed models, you say that there is a link also between pigment concentration in the leaves and on the one hand, the conductivity, and
Yes.
Positive with conductivity and negative with sediment phosphorus concentration. And the positive correlation you explain by lake trophic status. So you take conductivity as a measure of lake trophic status, but would phosphorus in the sediments not be a more reliable indicator for trophic status than conductivity? But that might be important in the interpretation because the sign of the correlation is completely opposite.
Yes. Well, apart that the correlation with phosphorus was not very much significant. I mean, it was quite borderline, but we also find that in the water, the phosphorus wasn't so high, even if it's clear that it's a eutrophic lake and there are so many algal blooms. So we thought that it would be interesting to look at phosphorus over time and not just have a single picture of the phosphorus in the sediment and in the water, because it may be very, very fast recycled by algae. So we thought that this may be the reason for these low phosphorus or negative correlations with phosphorus, but actually could be a proxy for the high bacterial and mainly algae activity.
Okay.
So
Room for
perhaps
one more question.
Oh, all right.
Yeah. No additional question. Luc, you still wanted to ask one?
No, I was going to start to take over. Sorry.
Ah, you want to take over? Okay, that's good. Go on then.
So I needed to introduce the third speaker because there's a few that Steven is involved in. So but Steven, I did of course not do my job properly. So I did not check the questions and answers section. So if you would know that there is a question, can you please mention?
No,
it?
there is actually no more on the platform. So there was
okay.
only one person who did that, and for the rest.
So it was not me being not doing my job. It was me being effective, not checking it because it was not.
You were just lucky.
Yeah. Okay, it's a pleasure to introduce to you Kimberly Lemon. So by the way, Kimberly, I found your video really beautiful and so very well done. So you can give your 2-minute speech and then people can ask questions.
Thanks. Okay, so I'm gonna share my screen. Okay, so just to give a little bit of background, so there's populations that lack genetic diversity that occur across the landscape, and this can occur for both natural and anthropogenic reasons. And what's interesting about these populations is that some of them can be quite successful, even in the face of environmental change. And this is contrary to the expectation that standing genetic variation is necessary for heritable adaptation. And so some mechanisms have been proposed that could introduce heritable phenotypic variation into populations. But what we're really lacking is an experimental test of if phenotypic variation can be produced, which is heritable, the extent to which it can, and the timescale of which this occurs. So I wanted to look at this question of, can heritable adaptation occur in the absence of initial standing genetic variation within an ecologically relevant time frame? And to do this, I had a little experiment with the microzooplankton Brachionis caliciflorus, where I exposed it to a culturing environment that favored phenotypic variance with elevated population growth rates in both high and low phosphorus conditions. And I had replicated populations, and I had 2 different clones, which I tested this in. And then at 2 time points during my exposure experiment, I ran these populations in a common garden to look for heritable adaptation. And so when we quickly summarized the results, what we did see was that in one of the clones, that we had a heritable adaptive response, which you can see well here. But what's really interesting to this session was that there was variation between the clones, so that we saw intraspecific variation in the capacity for such a response. And this is a cool kind of starting off project, which gives us a glimpse at the potential for populations to adapt to environmental change in the absence of standing genetic variation.
Thanks a lot. Are there questions on this? If not, but people can think further about it, I have at least one myself. Kimberly, you sort of, we normally would make a distinction between genetic variation and non-genetic variation, and you did it a little bit differently in the sense that you say, well, it didn't start from genetic variation. So that 2 options open, either it's due to mutations or it's epigenetic, I would guess. And so my obvious question is, which of the 2 do you think it is?
Well, so we don't have, we haven't done full genome sequencing, so we can't say for sure. But because we have 5 replicated populations, we saw similar patterns throughout these replicated populations, we think it's unlikely that it's a genetic mutation, at least in the kind of traditional random SNP concept that we would normally think of. There are possible alternatives, such as environmentally induced transposable elements, but those also have an epigenetic link to them as well. So we think it's likely an epigenetic mechanism, but we can't say for sure.
Okay. Ali, did you have a question?
Yeah, I was just curious if from this very short experiment, did you see any evidence for any kind of trade-offs due to this?
Okay, so we ran them in a common garden, and so the population that, or the exposure history that showed the adaptive response, when in the alternative environment, it did not perform worse than the populations that were from that environment. So in that sense, there's no obvious trade-off. However, the alternative environment is a very, it's a very good environment. So it might not be so obvious, these trade-offs. We're just not challenging it in another way.
Thanks.
So we have a question in the chat. So Miriam could ask it herself, but I'm just going to mention it. So first of all, thanks for the great talk. Would you expect this process to be important in the rapid variable environments? For instance, thermal fluctuations or something like that?
I think it really depends on the scale of the generation time of the organism of interest to the rate at which the environment's changing. And so there's very specific conditions under which you'd expect these epigenetic mechanisms to be relevant. And these are usually when the environmental change occurs for longer than a generation, but so it wouldn't be so rapid of within a generation. So it definitely has to be over a generation and has to be predictable from one generation to the next. You're welcome.
Okay, any more questions? Then if not, we should actually move to the next talk, which is on work by Chuajing Zhao, but Steven is going to present it. It's on rapidly evolving zooplankton in the salinizing world.
Okay, so as most of you probably know, salinization is a problem that becomes worse and worse for a lot of freshwater organisms all over the world. And it's important to realize that stress by salinization is not only due to osmotic stress, but also due to the toxicity of ions that make up the salt. And so in this experiment that was performed with Shuang Zhao and Libin Zhu, we tried to kind of see how rotifer populations, freshwater rotifer populations, are able to rapidly adapt to salt, but also to what extent adaptation to one type of salt may also confer or may not confer the ability to deal with other salts. And so we did a laboratory evolution experiment. We took then genotypes from each of these replicate populations being exposed to control conditions or salinized conditions. And we exposed them in a common garden transplant experiment to 3 different types of salt. Sodium chloride, which is actually the salt under which they evolved, and then calcium chloride and sodium sulfate. And it was not just a normal common garden experiment. During this experiment, we continuously measured population growth, and we continuously increased the salt concentrations of these salts up to the level that population growth became 0, population growth rate. And then we calculate the osmolarities, measure of osmotic pressure. And we will talk about 0 growth osmolarities here, and that is very convenient because then you can compare the 0 growth osmolarities between different salt types. And so in short, what we found was that populations adapted to sodium chloride were able to deal better, had higher zero growth osmolarity when confronted with sodium chloride itself. It's just to adaptation to that sodium chloride, but also when they were confronted with calcium chloride, but not when they were confronted with sodium sulfate. Now, regarding to the observation that they are better able to deal with calcium chloride, you could say, yeah, well, they got adapted to the toxicity of chloride, so that's why they also do better in the calcium chloride. But a more detailed investigation actually suggests that it's not only that, but at least partly also that the adaptation to sodium chloride allows them actually to be better able to deal with calcium toxicity.
Thanks a lot, Steven. Any questions on this? Perhaps I can start first again. First of all, I really loved the design of that selection experiment. I really thought this was very clever. At the same time, I sort of was thinking, given that these rotifers can adapt so fast, would you actually be influencing the end result by changing this? Because at a given moment you go below the growth rates and then you decrease the salinities again. So imagine that they would adapt very fast. Would that actually influence your results, or is it safe? Again, I found it very clever, but I was just wondering, sort of.
Yeah, I mean, well, first of all, okay, let me think this. So if you yeah, first of all, the changes you make, like increasing and decreasing and so on, is all the time on 1 day time interval. So this is, of course, very short compared to the time they had actually to evolve to this experimental treatment in the evolution experiment. There we talk about multiple generations with also sexual reproduction. So I think it's very unlikely, although they have a short generation time, the generation time is still more than a day. I would be very surprised that by just going up and down a little bit in order to get to that zero growth, that this would so substantially change the genetic composition of the population that
and it would be impossible to get a selective sweep, I guess.
And I don't think so. No.
Absolutely. Yeah. So do you have any idea why calcium and not sulfate?
Yeah, that's interesting. I think it's actually adaptation to sodium, and sodium and calcium are probably kind of similar in their effects. It seems from our experiment that calcium is actually more toxic than sodium, but adapting to sodium may help them already to get a little bit better, get along with calcium, and it has to do with pumps also, because the sodium and calcium pumps, somehow, I don't know the details of the chemistry behind it, but there is a link like between sodium and calcium pumps. So it is possible. So sodium intoxication also, so physiological, how would I say, dealing with sodium, too high sodium concentrations also involves calcium pumping. And if you and the other way around, and probably there is some link there in the process, in the mechanisms that result in such.
Thanks. I think Pedro had a question.
Yeah. Thanks, Steve. I was just curious. Often when I see experiments like that, is it worth to do the reverse? Once you adapt them to a high, you revert that process to actually get a better understanding.
Yes, absolutely. And it's very related also to the question that was asked to Kim regarding the trade-offs. So also here, we didn't find any trade-off. So the populations that had adapted to sodium chloride were at least as good dealing with freshwater conditions as the ones evolved in freshwater conditions. And again, it's a benign situation. So I started doing experiments with students now to see actually if a trade-off would not become apparent if you confront them with other kind of stressors. So it could be, for example, that the population adapted to sodium chloride can be better able to also deal with calcium chloride because of the specificity of sodium and calcium. But for example, if you would expose them to other stresses that are clearly different, like temperature or pH or so, then maybe the trade-off will reveal itself, and only then it also becomes relevant to do the opposite experiment. Otherwise, there is no real reason why you would do it, I think.
Luke, is there time for another quick question?
Very quick, and then Ali.
Okay, so one of the things that's happening here in Canada a lot, and we've been studying, I mean, we, meaning Canadians, not me, but meaning my taxpayers going to do a lot of study there. It's, we use a lot of salt to, on roads here, but it's a pulse, right? The salt is used more in one time of the year than the other, and there are different effects or whatever. And so I was trying to think whether this sort of experiments, they reflect the conditions in which populations are exposed, because the selection is not always there.
No,
yeah.
No, but we don't understand a lot of mechanisms of rapid adaptation of populations in nature. And this experiment is not to say, okay, in nature population. So it's a proof of concept. And of course, there's a lot of other context contingencies that may may determine the outcome of adaptation. And I think it's just a start. I think we definitely have to move on doing experiments that become more realistic. But this is a first.
Yeah, I was just curious.
So final question by Ali. Or should I mention it, Ali? Yes. So Ali asks you whether you would expect the salinity threshold where it stops, basically.
If I expect a salinity threshold, in what
sense? They can't
sense?
it up anymore.
Yeah, of course. And this is actually probably what we more or less reached already in the experiment, because here in my pitch, I couldn't explain it, but in the movie it's explained. We push them through alternative cycles of sexual reproduction in order to generate enough genetic diversity due to recombination. But then you see that at a given moment, the populations really don't do well anymore. And I think then we reached like the maximum. But of course, you should keep in mind that the number of initial clones is very low, and that real populations have, of course, many more genotypes. So it could be that that we make an underestimation of the adaptive capacity of natural populations here.
Yep. Thanks very much, Steven. So then we move to the next speaker, Libin Zhu, or Zhou. I'm sorry if I mispronounce your name. You can start by saying it yourself, and it's about macroevolutionary adaptation to stoichiometric mismatch, which reduces the cross-tolerance of consumers to salinization. And the floor is yours.
Is my screen?
Not yet.
No, not on screen. I think this is so I can see it.
Now it's coming, yes.
Okay, thanks, Luke. Organisms in nature are facing with increasing numbers of stressors, and such a general prediction of interactive effects of multiple stressors on organisms is very difficult, and it especially requires the consideration of evolutionary history, because increasing evidence have shown that evolution is able to occur at ecological time scales. So it is very important to see how the evolutionary history of organisms would affect their ability to cope with multiple stressors simultaneously. So for this study, we especially interested in how the effects of food phosphorus limitation on a rotifer species Brachiano classiflorus would be affected by their adaptation history to salinity and vice versa. In addition, we would also like to see how the interactive effects of food phosphorus limitation and salinization would be affected by their adaptation history. So for this, we experimentally harvest 3 types of rotifer clone lines, which are low phosphorus adapted clones, saline adapted clones, and non-adapted clones. And we performed a full bacterial population growth rate experiment to track the population growth rate of each type of the rotifers under each food treatment. And here comes the results. And in the graph, the circle dots means the growth rate, population growth rate of the low phosphorus adapted clones, whereas the triangle dots means the saline adapted clone lines. If the value of each dot equals to the gray line, the value of the gray line in the graph, which means that the population growth rate of the adapted clone lines has no difference between from the original clone lines. So it is interesting to see here on the high phosphorus food treatment, the LP adapted, the low phosphorus adapted clone lines have significantly lower population growth rate than that of the original clone lines, which means the adaptation to low phosphorus foods decrease the ability of rotters to cope with increased salinity. Under low phosphorus food treatment, if we see here, there's no differences between the original clone lines and the saline adapted clone lines, which means adaptation to salinity does not affect the ability of animals to cope with food phosphorus limitation. And on the dual presence of the stressors, we could see here both types of the clone lines actually have significantly higher population growth rate than the original ones, which means that the adaptation to one stressor, either food phosphorus limitation or increased salinity, still benefits animals under dual stressor conditions. That's my talk. Thanks.
Thanks very much, Libin. Any questions?
Yes, I had one. I was just typing it. So it was very interesting about the trade-offs, how one factor caused the trade-off apparently, but adaptation to the other factor didn't. So why do you think?
So we didn't experimentally investigate the mechanism behind, but we do have some speculations, let's say, or hypotheses, because we assume that actually the adaptation to low phosphorus actually have two types of mechanisms. One is a better ability to cope with phosphorus limitation, right? But on the other hand, it may have some it may have benefits. The adaptation history process may benefit animals to cope with excess carbon for the animals. So when the animals are facing with increased salinity, they may need some energy or carbon to cope with the osmotic pressure or to deal with the toxicity of the animals. So the low P adapted animals may have a better way to exclude or excrete the carbon, whereas the non-adapted animals may be better to use this kind of carbon to deal with osmotic pressure or toxicity of the ions. So that's kind of trade-off. Whereas the adaptation to salinity do not involve such kind of mechanism to let them to better cope with low phosphorus conditions. So that may account for that kind of result. Yeah.
And do you think that this would be universal across zooplankton, or this is just something we'd expect for rotifers?
Well, actually, we do not have some clue, but we speculate that because when organisms facing this low phosphorus condition, high CTP ratios, which means in any way they have to cope with excess carbon in their food. But the rapid adaptation to low phosphorus is not specifically for rotifers, but also was found for Daphnia or other organisms. So this, I think, for at least for aquatic zooplankton species, this result can be applied for such kind of organisms, I think. Yeah.
Okay. Thanks.
Thanks. Yeah, it just strikes me that there is this wonderful parallel between your study and then Kimberly and then Pedro's talk. I mean, we always think that we can explain things, or we will be able to explain things when we are studying like adaptation or species sorting and traits and so, but there is a lot of contingency in these adaptational things themselves. I mean, like different clones that Kimberly worked with give different results. And in your case, the order in which you expose your animals to stressors actually also makes a difference. So it's really intriguing, but also a bit worrying, perhaps. In the meantime, I have a question from Quentin Struck. So do you think, I mean, he just wants to make it a little bit more complex, I guess. And so he asks whether you have any idea what that would give for other stressors.
Well, yes. because I also performed some experiments with using like temperature and the food quantity. So indeed, some kind of interactions. For example, like if you face if you combine the food quantity and the temperature, increasing temperature, let's say. So under high temperature conditions, they need more. They have like higher metabolism rate, I think. So then they need more energy. So on the high conditions, on the high like temperature conditions, they may need more food. So the results are still in we are still analyze the results, but I think in the coming future we can give more clue on this. Yes.
Thanks. And there is a last, we have to move on, but there is a last interesting, intriguing idea of Miriam. She makes the parallel to low, fasting diets, in the sense that it's healthy because there is less free radical products. Could that be related to here, that you actually make your animals healthier because you keep them on a low phosphorus diet?
Yes, but yes. So actually, these animals are like lab cultured animals, and of course we selected some like we initially we do not want to some biased like selection on the animal like animal clones, because we have a clone bank in the lab. We just randomly selected some randomly selected some clones from our like clone bank. So I think in this case, so it can be representative, at least in the lab conditions. But of course, like it can. I think there's some like less representative for the nature conditions. But that's laboratory experiments. So I think, yeah.
But did I understand well that Miriam asked about whether low phosphorus food may be better for the rotifers? Was that?
No,
maybe I can explain. There are several experiments from bacteria to human that the low food makes you healthier in a general sense because of reduced free radicals production and also prolongs the longevity, you know. And yeah. I don't know, but it is from bacteria to Daphnia and monkeys and human. I think some years ago it was quite a fashionable topic. I was thinking that maybe because of better health conditions under the low nutrients could have helped the
But from all of our experiments, sorry, I take a little bit over Libin, but I think it's important to make that point here, that the low phosphorus food clearly is inferior food quality here. We see that time and time again. When we like do experiments with low phosphorus food, the rotifers clearly underperform very clearly compared to high phosphorus food. So, so I think I don't think, at least there may be such effects, but they don't compensate for all the other negative effects associated with phosphorus limitation.
You mean stoichiometrically?
Yeah.
Food. Okay, I understood the experiment as like the low food provided, not in that sense. Okay, then it makes sense.
Yeah.
We have to move on after this excursion into the rotifer world, which makes me gets me the scary idea that there might be actually more rotifer talks in this session than Daphnia talks. So that's very worrying, Steven. So please stop doing that. And so we can now move on, and Steven can take over again. Thanks, Steven.
Yeah. Okay, then is my phone, my microphone on? Yes. Okay, then we will move on to Lien Govaert. Lien, are you there?
Yes, I'm here.
Okay.
Okay, that should work. So hi everyone, I'm Lien. So I'm a group leader at the IGB where Luc is the director from, but today I'm going to present some still of my work of my postdoc that was in collaboration with Frank Pennekamp, Owen Becci and Florian Altermat. So basically the main aim of my of my study was to investigate how multivariate traits change in function of the abiotic environment, as well as the presence of other species. So, and here we clumped it basically into different species richness treatments. So basically we had a full factorial design of 6 different freshwater ciliate species. We had 6 different richness treatments where we basically had all possible species combinations. And then these were replicated along 6 different temperature treatments. So we had a lot of unique combinations. The experiment was run for 57 days, of which 26 time points were taken. And so basically of each of these microcosms, we take little videos, and then, well, I don't know if they are little, they're actually quite heavy. We take videos, and then those are processed with an ImageJ and B-movie art package. From those videos, we can subtract some data, as well as the density of our species, and also do species identification. And then I use that data to basically construct multivariate phenotypic trajectories, where we then look at specific properties of these phenotypic trajectories and compare where they, whether they differ between the temperatures and the richness treatment. So in my talk, I put forward some expectations, and then I also evaluated them. So I won't go through detail to them anymore, but basically what I want to say also, maybe as a follow-up on Luc's comment earlier, in my case, I think also maybe the species combination, so which species were together, might also actually have an influence on the patterns we see. And so this is basically one of the next steps I will take in this study system. Okay, and then I'll stop sharing.
Okay. Thank you very much, Lynn.
It's not a question, but I just had a comment. I never even knew that microbial surveillance was possible. So that's really amazing. Congratulations.
Thank you. I did not design this, but I mean the video, but it's super, it's really useful.
Pedro?
Yeah, Lynn, good to see you. It's funny, last person I saw outside of Canada was Florian in Iceland 5 days before the lockdown. I just came back from a meeting there. We had a good conversation about your work as well. I was wondering, how do you, so when you think about a phenotype, right, there are trade-offs in there, there are trajectories, but there are, for instance, correlations that you would think in terms of, for instance, speed and body shape and things like that. How do you, and then there's variance in that space, right? So how do we put a theory for coexistence, for instance, or even thinking about richness, or that you take into account the variance in that space, but at the same time, the mean phenotype that is being selected. So what's being selected? Is it diversity or the mean phenotype? And at the end, as I don't know, I'm assuming you saw my talk, but you have those two contrasting variance mean effects that, you know, some organisms could interact more in the mean space, and some could interact more in the variance space within the same community, right? So how would you go even think about separating those two effects?
Okay, I did not think about that yet, but I must also say, like, a follow-up study on this. So here we just look at multivariate means, and obviously it's very straightforward to go and look at covariance matrices.
Yes.
So that's our next step that we want to tackle, but we felt when starting to do analysis on this huge data set, it becomes very fast, very complex. And so we now first focus on the mean trait changes, then we'll go to covariance trait changes, and I think then maybe we can answer a bit more the question that you raised, how they differ. But then also, like, now we lump everything into these richness treatments, and I think that specific species combinations might actually also have a huge effect where maybe more similar species will compete more strongly. And so there's also something that we have to find a way on how to analyze. And then you think we have a huge data set, but I think for most species combinations, we maybe have only 2 or 3 replicates, which reduces the statistical power a lot to start looking at specific species combinations, I think.
Yes, okay, very good, thank you.
I have a question to Lynn.
Yes.
Very nice talk, Lynn, and of course the approach is very interesting. Probably you dealt with this, but I was wondering whether and how did you remove any ontogenetic variation in those traits, right? Because if you are measuring through time, how do we know that this is really like phenotypic plasticity and not just the animal growing, right? And then a new generation coming in. Do you track the same animal through time? So I think the video was just a bit too short. I have to watch it again. And maybe it was better for a longer talk, I don't know. And I was going also to ask probably what would be Luke's question. How do you think that evolution-mediated priority effects to the temperature gradient could alter the trajectories of species interactions along the richness gradient? So if they just enter a little bit earlier on those different temperatures, they may get stronger competitors in relation to the next coming species, and this might also affect their phenotypic interaction. So I think this might also play a role. And yeah, and I have other questions, but I will not make because of time, but very interesting talk. Thanks.
Yeah, so maybe to answer your first question, we cannot, well, we can track individuals on this video, but we don't track them throughout the entire experiment because that would mean we need a video inside of a microcosm tracking the individuals. That would be very ideal, but it's quite, I don't know if it's already possible or not. So we just take, I think the first 3 weeks, maybe they took every 2 days a sample, and then they did it once a week or twice a week. So we don't, so I think we don't like, I also don't know how much ontogeny or ontogenetic effects are in these ciliate populations, how important they are. The thing is that we cannot disentangle plasticity or genetic trait change because there's no common garden done at the end. So it's all phenotypic at this point. Then for evolution-mediated priority effects, so normally what they do at the start of the experiment, before inoculating the microcosms, they just pour the species together, they mix them, and then they sample. So there could be some stochasticity there going on, but I would think that you try to homogenize it this way as much as possible. And normally all the cultures are also kept at 20 degrees. So they should be, like it could be that a species prefers a higher temperature more, and then it takes off faster, but normally they are always kept already for quite some time at 20 degrees. So I would not expect pre-adaptation of the species to the higher temperatures or colder.
No, yeah, sure. It was more like a comment, or how do you expect this would play a role if you had
I
like
think
a
it
more...
would be a huge effect, no?
Yeah, so maybe a next step. Thanks.
Yeah, maybe.
Okay, thank you very much. I think we have to move on to the next speaker, and that is then Anna Nayeli. Anna Nayeli Lopez Rocha. Sorry.
Hello, my name is Nayeli. My work is called Variable Responses in the Demography of Brachiano savanensis exposed to Copperpodal chemicals. The aim of this work was to observe if there is an allelopathic interaction between cephalopods and calonoid copperpods in the demography of rotifers. So when we know that zooplankton species can interact not only through a traditional way like predation or competition for resources, but it also has been found that copperpods and rotifers in their life cycle, it's very associated with infochemicals. And both groups are very common in freshwater lakes, and they have strong interactions. Copperpods, cyclopods, and calanoids compete for resources with rotifers during their life stages, and in the case of calanoids, they are adult in their adult stage. They also consume the algae. Sorry, what screen I'm sharing? I'm not sure. Well, the species that they use come from a Lake Xochimilco. I used two species, the calanoid Actiniopterus dorsalis and a Mesocyclops, there is a cyclopoid in the Demography of Brachiano havanensis. We use a life table experiment, and that was counted every 12 hours, and the original cohort was followed till the last one died, and the neonates were removed every 12 hours, and they were counted. We calculate a life table variables. Well, these are the life tables that we calculate. For the we found that calanoids, the copperpod calanoids have a strong effect in that. With that I mean that the calanoid copperpod had an effect in almost every variable, like the average lifespan, the life expectancy birth, the growth rate, net reproduction rate, generational time, and population rate increase. While the Mesocyclops conditioned media did not have from females, it did not have any effect in any variable, and in the male conditioned media, we did find some effect in the survivorship parameters and in the net reproduction rate. That would be all.
Okay, thank you, Anna. Any questions?
I had one. So
Yes,
I'm sorry, I wasn't able to watch your presentation in full. So I'm assuming that these effects are negative.
Yeah.
On the root. Yeah.
All copperpod treatments have a negative effect.
Yeah.
Against
and usually when it's really nice to see a freshwater work with copepod allelochemicals on any organism, actually, because this is usually done in marine systems. But when we see it in marine systems, we see it's usually an inducible temporary effect. What do you think would happen if you place these rotifers back into a allelopathic free environment? Would they resume back to their original life table condition, or would you expect different long-term effects?
The effect that is usually observed is through generation. Usually, so if a rotifer is currently exposed to the Copperpodal chemical, and then the next generation is not exposed to, it'd be possible that the next generation will not have the same changes. I'm not sure if I'm expressing it well.
Yeah, I think so. So you wouldn't expect any kind of maternal effects, for example, from the
yes,
exudia?
Maybe, but I'm not sure. That has not been proved if there is an effect. I know that for some like Asplagna, that a predatory rotifer, did have some maternal effects, because when you observe the next generation, the Calyciflorus have longer posteral spines, But if that generation lacks of the presence of the splagna, the newborns will can have a shorter spines. So you can say yes, there is a maternal effect if the exposure continues over time, but it also cannot be. It can be the two ways.
Yeah, so that might be an interesting future direction for you if you have the option.
Yes.
I also had a question so you find, I don't exactly know anymore what kind of copperpot, but there was a copperpot who had quite a strong negative effect on the gross and net reproductive rates, but not on the intrinsic population growth rate. So what do you think could be the implications regarding the demography of the copperpot populations?
Can you repeat it again? I don't
Yeah,
understand.
so the gross and net reproductive rates are mainly important if there is actually, if you have a stagnant population growth, but if there's strong competition, then it will be the egg production that will be very important, lifespan egg production. Whereas if you have a very dynamic environment with strong population growth, still way below the current capacity just because of predation or whatever, then intrinsic population growth rate is going to be by far the most important, determining the demographics. So I find it striking that for one kind of trait, you don't see an impact of the copperpots, and for another traits, you see it. And these two types of traits have very different impacts depending on the context of the demography of the population. So
well, for the demographic, these demographic variables, it's reported that not every variable is sensible to allelopathic infochemicals. In the case of strong chemicals like drugs, the generational the population increase rate is a strong indicator, but because of the infochemicals are not an adverse chemical, how can I say it? They are not toxic. The effect is I'm stuck.
Stephen, may I answer
sure.
the question?
Yes,
Yeah,
yes.
The point is like that there are many life history variables, not all are all sensitive equally to the stress. Okay, number one, the rate of population increase is a product of survival too. It is a product of survival and reproduction. If an organism survives for longer time without reproduction, the rate of population increase is low. Okay, so in this variable, the rate of although reproductive output in terms of gross reproductive rate, net reproductive rate may be statistically significant, but the rate of population increase doesn't show because the animal might live longer, but per day the reproductive rate is low. So this is a product. That is why life history variables are not a single variable should be taken into account when you are evaluating the stress.
Okay, maybe we can discuss that later again.
May I ask Ali and Andros to inform us? I mean, we will get in trouble to meet 4 hours as a deadline. Is it okay to go beyond it, or is there anything else being scheduled that we have to get to?
Yeah, I think Ali can answer this better, but should be around midday, I guess. But yeah, so perhaps we can go a little bit further, right? But at one, yeah. So go on, Ali.
So when is this early career workshop starting?
Yeah, so right now it's 11:40 in Brazil, and the scheduled end of this session is at noon, and the early career is at 1. So in about 1 hour and 20 minutes.
Okay, well, we can go, say, 20 minutes beyond time.
Yeah, sure.
I think we should try
Sure.
to keep it within limits, but on the other hand, we also want to give the opportunity to people to have a bit of decent discussion.
Yeah,
also.
I mean, yesterday there were sessions that went over by half an hour. So if there's a lively discussion, that's fine. I might have to leave because I have to start organizing the early career, but when I leave, Andros should be the main host. So I hope that's
fine. But that's fine. Just to know, thanks very much.
Okay. Okay, thank you very much, Anna. And then now we will move on to Helen Asagilt.
Hello to everybody. Can you hear me?
Yes.
I will share my screen now.
Yes, thank you.
Can you see it?
Yes.
The main aim of our study was to compare the trophic structure and carbon sources of fish communities in Amazonian black and whitewater lakes during the high water and low water stage. And this presentation, this is actually preliminary results of our research, and I have a chance to give you overview and first results of the high water stage. And we had two study lakes. One was Lake Prato, which is Blackwater Lake, and this in the watershed of Rio Negro, and another lake is Whitewater Lake, Lake Chanahuaca, which is connected to River Amazonas. And to have the overview of the full fish community, we used Nordic type gill nets, and these nets had mesh sizes from 5 to 55 millimeters to catch both the small and large fish. And we had floating and sinking nets, and the nets were put for both open water biotop and to littoral. And we had only chance to catch the fish during one day and night. So this is snapshot research. And our results showed that comparing these two lakes with the stabilizer top analysis, we saw that the sources available for the fish communities in Blackwater Lake are much wider compared to Whitewater Lake. And this was also seen considering the different fish feeding groups. And the main take-home message maybe from this very short research could be that use of free isotopes in case like we had, only one chance to take the samples, is definitely advantage in studying fish communities and food webs, for example, compared using only two isotopes, carbon and nitrogen. Thank you.
Thank you very much. Any questions?
I may have one. Thank you very much, Helen. I was struck in your video, I may have misunderstood, but I was struck that there is basically no species overlap at all between those two lakes, which was really striking to me because they're at least sort of in the same catchment, at least connect catchments. So is then the ecology of the lakes so much different? And if so, how do we have to deal with those isotopes? Because it might be that it's simply totally different because our total settings are different, or because the fish behave differently.
There is actually, yes, thank you very much for these questions. The lakes are really very different, and the fish communities were different. There were no similar species. So we are actually in the situation comparing totally different communities. One of the lakes belonged to the dark water and another one the white water, and they were different watersheds. So yes, this is quite intriguing questions how to solve this. At the moment, we just compare the isotopic results and try to find out how big are the differences in carbon use between these fish assemblages.
Okay.
Any more questions?
I have also a question. So you see regarding carbon, that blackwater food web is actually more depleted in carbon than the whitewater food web, and you relate that potentially to the consumption of methane oxidizing bacteria. Is there anything known about the relative or about differences between black and white waters regarding methane production? Would there be just, and that refers a little bit to what Luc also said, the systems are probably so different that
Yes, we assume that this methane effect to the food webs could be higher in the blackwater lakes, and in Amazonian areas are particularly known for high methane emissions. And this was actually one of our research aimed to find out whether there could be some effect from the methane carbon to the food web. In our home country, actually, we have started the research studying shallow lakes in terms of methane carbon in the food webs, and we were quite interested to see and compare the results to those lakes in Amazon area. And the results from this blackwater lake, especially for zooplankton, might indicate some contribution from the methane. But to be absolutely sure, we should do additional analysis, for example, fatty acids or DNA analysis, which are actually planned, and we collected samples from the another sampling campaign in 2019, which we had there and during the low water. So more results are coming soon, but at the moment we can just guess that this is effect from the methane.
But to generalize, do you think that there is like a systematic difference between black and white waters regarding to the methane production of the entire system, and that is then reflected in the isotope signatures of the entire food web? Or is it more that there is more consumers that are actually linked, that there's a stronger trophic link in one type of food web compared to the other one regarding to methane oxidizing bacteria?
Quite complicated question. I'm not able to answer this right now, but I think yes, in dark water lake there could be more systematic consumption from this source.
Okay. Any more questions about this talk? No? Well then, thank you very much.
Thank you.
Then we go to Rafael de Togni Guariento.
Hello everyone, can you hear me?
Yeah.
Okay, thank you. So I will share my screen here. Sure. Okay, so first of all, I would like to thank you all for having us, for organizing this meeting. I speak very slowly, so my apologies in advance. And we know that we all wanted to be in Natal, enjoying the sand, the sea, and the sun, but it is what it is. So very nice to be here and to see some familiar faces like Ali is right there. So thank you all. Our presentation here was conducted actually by my student. He's not even a master's student, he's an undergrad student, Paulo Henrique Santos. He is on the audience. Hello. And will probably help me during the answers. So we all know that predator and prey might fluctuate in time. Many models show that. And earlier work by Martin Scheffer showed that fish predation can actually stabilize these dynamics, which is very important in shallow lakes. However, we know that zooplankton does not accept his fate of just being killed. We know that defenses might occur in these populations due to rapid evolution or due to plasticity. And our goal here was to challenge Scheffer's conclusions and challenge the Scheffer's work, saying that maybe fish will induce defenses, and this defense may counteract the effect of predation, on the stabilizing effect of predation on these zooplankton algae dynamics. We used a model here. It's a framework that was based on the Martin Scheffer's work. However, the attack rates of the fish, of the predation, was different for two different populations. So we have a population of daphne that has no defenses, and this population differentiates into a mutant population, a population with defense using a switching function that is common in eco-evolutionary dynamics modeling. And we also, so this predation rate, this attack rate is lower, is actually does not exist in populations that has a defense. But we also expect that having a defense will induce a cost in foraging time, a cost in energy allocation, any kind of cost. So those populations that have any defense will present lower efficiency in efficient conversion of algae into food. So they have a no cost. So the efficiency of the two daphne populations will be the same. A low cost and a high cost. So lower and lower value for the efficiency, the constant of the efficiency conversions, the parameter. So let's go to the results. Here is the, no, this is not the results. So no, here are the results. Okay. So this is the dynamics for the algae population. So what I have here, when we have a line, we have stable dynamics. When we have a dotted line, we have a limit cycle. And what we can see is that when predation decreases, we see that the population starts to fluctuate. It is especially evident in when nutrients are high. So we also have two levels of the resources here. When nutrients are low, we have a space here where even when nutrients are high, at no defense cost, populations will always fluctuate. It doesn't matter the fish predation. However, when the cost of the defense increases, so have low cost and high cost, we start to have stable populations for some fish predation. Actually, the effect of predation stabilizing the dynamics actually occurs even at low predation rates. So the conclusions here are that can we challenge the Shepherd's conclusion? Can we say that fish predation will not have a stabilizing effect if zooplankton manifests defenses? Actually not if we assume a trade-off that when defenses occur, there will be a cost. So in fact, maybe Shepherd's conclusions will occur even when zooplankton have defenses, because it will stabilize the dynamic through an indirect pathway by enabling defenses that have some kind of cost to growth or the fitness of the zooplankton population. So that's it.
Okay, thank you. Any questions? So you have a strong focus on the costs of the defense, but I'm not a modeler. But did you also study the effect of the efficiency, the efficiency of the defense, and the potential interaction of efficiency with the cost? Because that interaction may also potentially determine the dynamics strongly. Or not?
I can answer this.
Yeah, that's complicated because Paulo may be trying to speak now. Paulo, you can answer.
Yeah, I can. Actually, the defense, the benefit from the defense impacts a lot on the dynamics, and it's something that we looked at our study. But we had to settle at some point, and we settled at we studied the literature, and we settled at a point where we think was reasonable to the fences from fish and other invertebrates at a reasonable point. And we focused our study really on costs, but it actually has a big impact on the dynamics.
Okay. Any other questions?
Yes, I may try if the Belgian internet providers do not kick me out again. So I was wondering, you are talking about cost. I did not really check the model. I mean, I honestly have to say that I didn't check it, but the cost was not implemented in terms of the grazing on algae, because that would be another way to look at it. Most of the effects of fish predation would result, other animals would result in hiding during the daytime and stuff like that. That would all impact grazing on the algae directly. So I wonder if you have explored that.
You are right, because if you look at the equations, what is being removed from the algae is not affected by the cost, because it's just affecting the growth of the daphne. So in fact, yeah, we could do that as well, and of course this could affect the results. This maybe some kind of direction that you can take going forward.
Okay, Ali had a question.
Yeah, first of all, I'd like to congratulate Paulo. It's great work for an undergraduate, and it's nice to see undergraduates here in addition to grad students. I always think it's nice to have a mix. And so I just wanted to ask, are you planning on testing this experimentally?
Paul, go ahead.
Maybe, actually, we have so many abilities for this model going forward. I think it opens a lot of questions, and we maybe are going to do something experimentally, but we are not sure yet because it's still too early to I think.
Okay, good luck. Sounds interesting.
Okay, thank you very much. We have to move on to the next talk, and that is the talk of Natalia, Cristina, Biera de Carvalho.
Hello, everyone. Are you hearing me?
Yes.
Okay. I'll just share my screen. I'll just show my first slide. So the title of my research is Temporal Dynamics of Phytoplankton in a Shallow and Mesotrophic Lake, and this study was part of my master's. So the phytoplankton community is very diverse and composed of different taxonomic groups, which include microalgae and cyanobacteria. The community structure is governed by different environmental factors and biological interactions, and the predominance of one group over another in a given aquatic system is related to certain ecosystem characteristics. We present results from a study performed in a shallow mesotrophic lake in Brazil, located in a karst system, which has been suffering from environmental degradation. In previous studies, this lake presented a permanent and stable dominance of Apidiops hasibolski, a potentially toxic cyanobacteria. And to follow up the dynamic of the phytoplankton community and verify the environmental factors that govern it, we performed sampling over temporal gradients. Water physical and chemical characteristics were evaluated, as well as the phytoplankton community through identification and quantification of organisms. The study reviewed alternation of dominance between cyanobacteria and chlorophyton. And differently from previous observations, the cyanobacteria species Apidiops hasibolski dominated the community only during part of the year. The water column stratification and mixing process observed in the lake may have favored the changes that occur in the species composition and dominance factors. Sorry, A functional traits approach used in the present study was important to understand the strategies adopted by each species. And environmental variation seems to have produced an alternation between R and K strategy species over a temporal gradient. Biological interactions like competitive exclusion, to which phytoplankton species are subject, must also have contributed to the changes observed in the community. And by focusing on the species' morphological and ecological characteristics, it was possible to better understand the response of the phytoplankton community in face of changes occurring in the ecological structuring factors.
Okay, thank you. Any questions?
Perhaps I can ask a very brief one, Steven. So thank you, Natalia, sorry, for this introduction. In your video, you mentioned this RK strategies.
Yeah.
And so I wondered whether you did any statistics on that. I mean, so you present that, but the question was whether you, through the season, for instance, or in any other way, did statistics to see whether there was a shift from R to K in different groups and whether those dynamics were different.
Yeah, we didn't did this. We just use redundancy analysis to correlate the biotical variables and abiotic variables, but we didn't use statistics to compare the K and R strategies.
Because it might just be interesting to just do that in addition to your description of the species, to look at how the traits change and, for instance, link it to R and K or to other things like just body size or something like that.
Thank you for the advice.
Any other questions? So do you know anything about the dynamics of the fish populations in the lake, and also regarding to predation on zooplankton?
No, we didn't study that.
Okay. Do you think this could also cast a light? Suppose you would have done it, do you think you would have learned more from it, or?
Maybe the predation over the phytoplankton could be something interesting to study, because we analyzed just the competition between the species of the phytoplankton, but I think the zooplankton species could also impact the community.
Okay, okay. And is there any like the fish, like, is there a continuous production of fish fry in the lake throughout the year, or is a clear seasonality in fish fry production? Because mainly the fish fry probably that are the main planktivores.
I think I didn't understand the question.
Yeah, well, in the temperate regions, you have this kind of fish fry hatching, and then you have like high predation pressure on zooplankton, but then the fish grow up and start switching to invertebrates and so on. So there's like a very clear dynamics, seasonal dynamics in temperate lakes, which may affect zooplankton population and then the impact of the zooplankton on the phytoplankton. Whereas in tropical lakes, yeah, it may differ a lot, although I don't know too much about it. So maybe, yeah. Do you have any idea about that and how it can relate to your
Alessandra,
observations?
could you help me? Alessandra is a collaborator of my study.
Excuse me?
Alessandra
Okay, we don't have data about fish population in this lake. It's a very small lake. So I don't think there are any difference between seasons. So temperature is not that it doesn't have a large switch from the rainy from winter to summer. So theoretically they are having reproduction the entire year. And so I think there may be a correlation with zooplankton that we didn't measure. But what we found that was very interesting actually in this lake, because we have been studied in this lake before, and there's a strong dominance of cyanobacteria, like 12 months per year. And especially when Natalia was doing her master there, we observed suddenly the lake turned so red, and there was this dominance of chlorophytes. So, and they produced a lot of carotenoids. So what we thought at that moment, we tried to find what's happening. And what is
I think there's a problem with the internet connection.
Yes.
Yeah. Okay. Look, can you write her that we don't hear, or Andros? Because it's a bit rude to just stop
we
her.
need to move on.
Yeah. Can you write the chat?
Yes, of course.
Yeah. Okay, thank you. Okay, then we move on to the second last talk from, I don't know how to pronounce it, Gleiche de Souza Santos. Okay. Okay, thanks for the help.
Can you see my presentation?
Yes.
So
anyway, so, the idea would be
Okay, also here there seems to be, unfortunately, I don't hear anything. Do you?
I'm not hearing as well. Gleice, are you still around?
Yes, I'm here.
Okay, all right.
Okay.
So you can,
can
give
you,
your presentation, please.
Yeah.
Okay, so our work is entitled Zooplankton Functional Diversity in Lagoon Ecosystems Impacted by Mining Tailings. So we compared zooplankton communities in 3 shallow and 3 deep lakes of Brazil. There are lakes really impacted by pesticides, eutrophication, and also mining tailings. And we hypothesized that functional diversity of zooplankton would be higher in shallow lakes because of the environmental complexity provided by macrophytes that improve zooplankton richness. So we predicted that macrophytes in shallow lakes should also improve functional diversity of zooplankton in relation to deep lakes. But our results showed a different pattern, and deep lakes showed higher functional lakes, and we suggest that maybe larger and deeper lakes provide higher, more niche opportunities for zooplankton that promoted a high functional diversity of zooplankton. Other features differentiating shallow lakes and deep lakes were some environmental variables. For example, shallow lakes presented higher total organic carbon, suspended particle matter, total phosphorus and nitrogen in relation to deep lakes, and also higher iron concentrations due to the presence of the mining tailings in this environment. So these characteristics should also decrease zooplankton functional diversity in relation to deep lakes that showed higher chlorophyll A and cyanobacteria density. So maybe these environmental characteristics also explained our results. And that's it. Thank you for your attention.
Thank you. Any questions? Yeah, Luc.
Perhaps just a small one. Thank you, Glacy. So in your video, you sort of emphasized this higher niche availability in deep lakes. Whereas I was then sort of wondering, what about the mine tailings? So now you indicate that might be this pollution effect. So would that not just overrule the other hypothesis that there is more niches? I mean, in the shallow lakes, the animals are just killed.
Yes, probably other thing is that in deep lakes, according to the literature, a lot of species can survive to predation because they develop mechanisms to escape of predators. So maybe escape avoidance should also promote this higher functional diversity in deep lakes in comparison with small lakes that organisms are more, let's see, concentrated in a small place.
Okay.
Thanks.
So when you, sorry, Andros, yes.
Oh yeah, I have a question. I liked your presentation in the sense that you went very much into detail in the basics of how you built those indices, right? The functional diversity indices. I think this is even good maybe to share with my undergrad students in the class. But my question was is more related to whether or not you included abundances or whether you corrected for richness difference among those lakes, because it might be just an artifact that you have more species richness in deeper lakes because you have more volume and then you have more abundance. So total N or abundance may affect richness, and that's why you have a higher or a bigger functional volume. So did you correct for this? Otherwise, I think it should be an interesting step to really remove this effect. Thanks.
Yes, yeah, deep lakes and have a higher abundance, as you say, and a higher richness too. And maybe this also explains our results. Yeah, you are right.
Yeah, so maybe after removing this, this effect, maybe you will corroborate your hypothesis, right?
Mm-hmm. So, yeah.
When you calculate functional diversity, you put several traits in it. Actually, these are all traits that are easy to measure, categorize, so that's what you put in. But of course, something like size or diet preference is a completely different thing than, for instance, the reproductive mode or the number, or how quickly they kind of like reproduce or so. So I wonder, like, why, like, what was the objective of your definition of functional diversity? Was it a response trait diversity, or was it an affect trait diversity? And if it was the latter, then why would you include things like, as I said, for example, I thought I wrote it down here, like traits like lifespan and escape capacity. Like, how would that affect the impact of the populations on the system?
Sorry, can you repeat? My internet's a little unstable.
Yeah.
And I cannot understand well the last phrase. Yeah. Can you repeat, please?
Yeah. So in your functional diversity, you put things like body size, which is a very important trait, because a lot of other things are associated with it, or the feeding niche, that is both a response and affect trait. But then there is other traits that you put in your analysis for calculation of functional trait diversity, for example, lifespan and escape capacity. But for me, it is much more difficult to envisage how this would mediate the impact of the population on the system, or how this relates to the other traits like body size and diet?
Because we predicted that species that is good in escape from predators, they can stay a more time in the ecosystems and developing a function in that ecosystem. So species that can be eaten easily, they will stay for a short time in that ecosystem. So, and that's it.
But there might be trade-offs, of course. It might be that the species is eaten more easily, but compensates that with an R strategy and is demographically kind of compensating for that. So
yeah,
traits all kind of somehow depend on each other, and
yeah, maybe.
Yeah, for me, it's not always clear like what people mean with functional diversity, and I just want to know like if you had like clearly in mind what you wanted to define with your functional diversity and why you choose these traits, and that was a bit what I was wondering about.
Yes, because we understood that these traits are important for lakes. For example, maybe if we study population of rivers, we should consider other traits. So in our set of traits, we consider that the time that species can stay in the ecosystems maybe can explain the functional diversity of zooplankton.
Okay. Okay, so thank you very much. Is there any other questions? Is there any more questions? Hi, Cristina. Hello.
Hello.
Hello,
everybody. Grace, congratulations for your research.
Thank you.
Which was done in a very important system, heavily impacted by bauxita tailings.
Yeah.
Yeah, it's a shame for us in Brazil, but now we are trying to recover it.
Yeah.
And a question, do you consider the migration of the zooplankton? That's a very important escape strategy from fish and also insect predation.
No, unfortunately, we don't have data about fish predation or insect predation, unfortunately, because this area started to be is studying now, yeah, and our recent studies in this area. Maybe we know that it's an important issue, yeah, because the systems are really complex, but unfortunately we don't have this data yet.
Okay. Okay, thank you very much, Glaise. And then we come to the last contribution, but the author wrote me an email apologizing for not being here. So I'm afraid we won't have a presentation and we won't be able to ask questions, except if there would be a collaborator from Chaparro Griselda here who could receive our questions. No? Okay, if not, then I think we are already over time. So thank you very much. And I think I very much enjoyed this session, and I hope you did as well. I don't know, Luc, if there's something you still want to add to this?
Yes, I wanted to also congratulate all the authors also for the very diverse. I think it was very nice. We were talking about trait diversity, and there was very diverse talks also. Before we close, I do not know whether Steven did his homework, but I didn't. And so I do not know who are the pre-PhD defense candidates, because we have to select best doctoral student presentation.
Yeah, but Coca send us a list.
We have a list.
So. I hope we can get it out of the list. So, but otherwise, maybe it's easy if everybody among the
I just wanted to
that
ask
they just send an email to me, and then I will share to us, and then we can discuss.
So please indicate whether you are a postdoc or not, so that we.
Well, whether you are undergraduate, PhD student, or postdoc, or what.
Okay.
Okay, so I also would like to thank you very much, Stephen and Luke, for organizing this session. I think it was very interesting. And I also have to congratulate you for keeping in time, although you had so many, because it was challenging. You had so many presentations. So thank you very much for you guys and to everybody who presented beautiful work here and nice discussions. So see you later. Bye-bye. Cheers.
Okay. Thanks for hosting us, Andres. Bye-bye.